Good evening, Year 11. Welcome to your Biology Paper 2 Trilogy revision. So we'll be going through the trilogy content first, and this will take us through to 7 o'clock. And I'll be coming with the separates only content from 7 till 7:30. Thank you. Over to Miss Hughes for Spec 5, Homeostasis and Response.
Right, so good evening, Year 11. So we're going to go through, this time, a slightly different approach. So we're going to do a mind map, first of all, on what we think the key parts to this spec are going to be for your exam, and then we're going to look at some exam questions and see how they link in. Okay, so we're going to start with homeostasis, and we're going to start with what the definition for homeostasis is. So it is the maintenance of a constant internal environment. So maintenance of a constant internal environment. Spell that wrong. Okay, now there's reasons that our body needs to maintain this constant internal environment. Okay, so it's needed so that our enzymes and our cells can function. Okay, so one of the ways we need to know for our exam is our blood glucose. So we need to know how our blood glucose is regulated within our body. So it is maintained, or monitored should I say, monitored and controlled by an organ called the pancreas. Okay, so pancreas is the organ which monitors and controls your blood glucose levels. Now, we need to know what happens within our body if they become too high or they drop too low. Okay, so the first one we'll look at is what happens when it gets too high. So the hormone which the pancreas releases is called insulin, and we need to know what insulin's role is within the body. So what it does is it moves glucose from the blood and into the cells. Okay, so that's its role within the body. In the liver and in the muscle cells, some of the glucose is converted into glycogen and is stored. So excess glucose is converted into glycogen. Okay, and then there it is stored. Now the whole reason of doing that is obviously to lower our blood glucose levels. Okay, so what happens when our blood glucose falls too low? Another hormone is released, this time called glucagon. Okay, so that is our hormone. Now this time, the glycogen which was stored is converted back into glucose. Okay, and when it's converted back into glucose, it is then released back into the blood. And obviously when it's back in the blood, the whole reason is to increase our blood glucose levels. Okay, so that's all we need to know. What happens when it gets too high, the hormone and what it does, And the same again when it gets too low, the name of the hormone and what it does. Okay, so linking into blood glucose, we're going to quickly look at diabetes. There are two types of diabetes. We've got type 1 and type 2. Okay, so type 1 diabetes is where your pancreas, remember that's the hormone that controls our glucose levels, monitors and controls our glucose, is basically the pancreas fails to produce any insulin. So if it's not producing insulin, then we can't lower our blood glucose levels when they become too high. Now, a treatment for this is to have an insulin injection. Okay? Type 2 is slightly different. This is where your body cells don't respond to the insulin produced. Now, this treatment is slightly different. So people who've got type 2 diabetes can treat themselves through controlling their diet, but also through doing exercise as well. Now, one of the risk factors associated with type 2 diabetes is obesity. So, the last bit of this topic that we're going to focus on before we look at some exam questions is the required practical that we think could come up this year, and it is the reaction time required practical, so it's the ruler drop test. Okay, so it's used to measure reaction time. So we're going to run through a quick method. So just to remind you, we have two people. So person A, they would be the one with their hand with a gap between their finger and their thumb. Okay, so they'll be sat down with a hand over the table and a gap between their finger and their thumb. Now their partner, so we could call them person B, will be holding the ruler just above their finger and thumb, so that 0 centimetres is level with it. Now person B drops the ruler without warning. Okay, and obviously person A will catch it. When person A has caught it, you then need to read off the ruler where they've caught it. So you could read off from above the finger and the thumb. And then obviously you would repeat again. Okay, so that's a basic method for the reaction time required practical. It's really, really simple. The other thing that they could ask you linked into this is how you could change or how you could improve this practical. So you could look at using caffeine to see if reaction time improves when someone has caffeine. You could look at changing hands. So that could be your dominant or non-dominant hand. Or you could even look at the lack of sleep and how that affects your reaction time. Okay, so that is all we're going to cover for this part of Spec 5. Okay, so if you want to make any more notes, you can go back and look at the video again, because we're going to move on to some exam questions. So you can see how they could test you for this part. So, the first one, so question 1, we've got 3 questions to go through. So this one's looking at blood glucose concentration. Okay, so it says the concentration of blood glucose in 2 people, so we've got A and B, is measured every half an hour. 1 hour after the start, both people drank a solution containing 50 grams of glucose, and then you've got a graph below which shows their results. So let's look at the graph first. We've got person B at the bottom, person A at the top, and you can hopefully see the line on the graph. It tells you exactly when they took the glucose drink, and along the bottom we've got time in hours, and then the concentration of blood glucose up the side. So the first question says, by how much did the glucose concentration in person B raise after drinking the glucose drink? Now for this question, please take your time to work out what each little square on your graph is worth, and do not go and just guess a number. Work it out. It might sound very easy, but you won't believe the number of people that just get this wrong all the time. So work out what each little square is worth. In this example, it's actually worth 1. Okay, so it's really simple. You just count up from where they took the drink up to where their blood glucose concentration has increased to its maximum. You could even use a ruler and rule across to the axes. It makes it easier for you to read it. Okay, so it's increased by 11 in this case. Okay, so that's all you would do. Take the time to work it out and don't just guess. Second part of the question. So a doctor suggests that person A has diabetes, so that's this person here at the top. Give 2 pieces of evidence from the graph to support this suggestion. So we're going to look at the graph for evidence to support this. Okay, so if we look at person B compared to person A, well, we can see that person A has a much higher blood glucose concentration at the start compared to person B. So that could be one of our answers. You can quote the starting figures if you want. So you could say that person B has got a blood glucose concentration of 88, whereas person A has got a blood glucose concentration of 115. You would only be able to accept the numbers, the comparison of numbers. So you need both numbers in your answer. We could also look at how much the blood glucose has increased. So person B, we worked out has increased by 11. You can quite clearly see that person A has had a much greater increase in their blood glucose concentration. We could look at how quickly the concentration has increased. Person A has got a much steeper or higher gradient here, so their blood glucose concentration increased quicker. So that could be a third possible reason. And then the last one, look at how quickly they fell. Person B's blood glucose lowered much quicker than person A. So there are lots of different pieces of evidence we could take from this graph. There's actually 4, but you only need to give 2. The last question says, give 1 reason for the fall in blood glucose concentration in person B shown in the graph. So why does blood glucose concentration fall? Well, think about what lowers blood glucose concentration, and the answer is obviously that they produce insulin. Okay? So, moving on to question 2. Now this one again is a blood glucose concentration question, but it's an extended answer. So this one was worth 4 marks. So it says, explain as fully as you can the reasons for changes in blood glucose concentration when a person ate brown bread. So I'm just going to highlight that bit there. When a person ate brown bread, is suggesting they've eaten something, so their blood glucose concentrations are going to be increasing. So we need to explain why that could be. Okay, so we're going to look at what happens when we eat bread. So in bread, we've got starch, and we know that starch is broken down into glucose. They would even let you have for this mark scheme, sugar. Okay? That obviously then goes into our blood. And from there, we need to use our knowledge of what we've learnt so far about blood glucose and what happens when it gets too high to suggest how our blood glucose would change. So we need to reference somewhere in our answer the pancreas and the hormone that it produces, which would be insulin. Okay, once we've mentioned that, we just have to say what insulin actually does. So we did it on our mind map. Insulin moves the glucose from the blood into our cells. You can say also in this answer, tissues and organs. You can also even say that the glucose is used in respiration. Okay, so that will get you 4 marks. The final question is linked into the required practical. So this one is looking at caffeine found in coffee. After a person drinks a coffee, information passes through neurons in the nervous system more quickly. Suggest a hypothesis for the effect of caffeine concentration on reaction time. So a hypothesis is something that you would want to test in a practicum. Okay, so it's looking at caffeine to start with, and it wants to look at reaction time. So how does caffeine affect reaction time? So you can create any hypothesis you want. You could say that the higher the caffeine concentration, the faster someone would react. Or you could even say the reverse. So the lower the caffeine concentration, the slower someone would react. Okay, so that's a hypothesis, something that you want to test. The final part of this question is you've actually been given a method for the ruler drop test. And the question says, suggest how this method could be improved to produce valid results. So all you would do is think about the practical, look at what they've given you in the practical, and how could we improve it. So student A drinks a cup of coffee. Well, if we just look at this sentence alone, we could have many different things that we could do to improve this practical. We could use a cup of coffee. We could also use decaffeinated coffee. And we could use it as a control to compare our results to. We could look at doing a double-blind trial. So that is where neither the student who's drinking it or the person who is giving the caffeine would know whether they were having coffee or not. You could leave the experiment for, let's say, 10 minutes before you start. So make sure that there's a time between when you drink it and when you test. Now this student here said hold the ruler above the student's hand. Well, in our method, I said hold it at 0 centimetres, so that's just next to the thumb and the finger. So you could mention here, have a starting position for the ruler. If you wanted to, you could look at different concentrations of caffeine. So you could see whether actually increasing the concentration of caffeine increases the reaction time even further. Now, quite a couple of the hard ones further down this mark scheme, you could even use just a caffeine solution, and that would eliminate anything else that may potentially be in the coffee that could affect someone's reaction time. And obviously, they've only done it once, so the obvious one that everyone always says is to repeat the investigation and calculate a mean. You can say repeat as many times as you want, just as long as you mention repeats. You could say 3, you could say up to 10. Totally up to you. So we're just going to take a short break now while we pass over to Miss Neresi, who is going to be doing Spec 6 revision.
Would appear I've put the wrong sheet in front of the camera, guys. Apologies. This is the Spec 6 one. Obviously, like Miss Hughes, I can't go through everything that might appear on Spec 6. There's quite a lot. However, if we look at what came up last year and what didn't come up last year, I've highlighted in orange the bits and pieces that didn't come up last year. So we're going to have a look at mostly inheritance, some evolution, genetic engineering, fossils and extinction. These are the points that didn't really come up in much detail. When I say genetic engineering, I mean the process of genetic engineering, because advantages and disadvantages did come up. So we spent a lot of time in lessons looking at the keywords associated with inheritance. If you do not know these now, you're in a bit of a poor condition or situation, because this didn't really come up last year, so fingers crossed it'll come up this year. So the key terms, mine isn't going to look half as neat as Miss Hughes, but we're going to go for it. So first one, homozygous. We need to know what homozygous means. If you look at I can't speak, first part of that word, homo, it means the same. If an individual is homozygous, it means that the alleles are the same. For example, if a person was big B, or two little b's. The opposite to someone being homozygous is heterozygous, and this means that the alleles are different. So if we look at heterozygous, the first part of that word, hetero, means different, so the alleles for the individual are different. E.g., big B, little b. That would be an heterozygous individual. I've said alleles twice now. Hopefully by this point in year 11 you know what an allele is. An allele is a different version or variety, different version of the same gene. So you get two versions, one from mum, one from dad. Okay? Linked to this, we need to know what the different alleles could be. So we could have a dominant allele, if I could spell, or we could have a recessive allele. A dominant allele, the characteristic or phenotype is always shown. So a dominant allele is always expressed. A recessive allele, you need two copies present to show the condition. You should hopefully know which one is shown with which type of letter. A dominant is always a capital letter, recessive is always a lowercase letter. Okay, these are some of the key terms. Carl Linnaeus, I enjoy that comment. Yes, I'm far superior to Love Island. I'm enjoying these comments so far. Other terms that we need to know. We need to know what phenotype means. Phenotype is the physical characteristic, and that might be brown eyes. That's a physical characteristic. The genotype is the genetic makeup, and that is always two letters. If you are asked for a genotype, it means which two letters, so it's always going to be two letters. I can't say that enough. So for example, someone with brown eyes, their genotype could be big B, or could be big B, little b. I'm going to show you why that is possible now. These are just some of the terms that you need to know, and you need to be able to do these if you're going to be able to work out Punnett squares. So you might have a situation where you're giving an exam. There are two people that are having a child together. They've both got brown hair. What is the possibility of them having a child with blonde hair? So if we look at these, a person with brown hair, because brown is the dominant allele, you can tell that even if you're not sure by how many are brown compared to blonde. The trait that appears the most is going to be the dominant trait. So for example, if we had a mum with brown, we'll go brown eyes, and we had dad with brown eyes, and they managed to have a baby with blue eyes, this should tell you that someone or both of them have at least one recessive allele, because to have blue eyes, which is a recessive condition, the baby's genotype must be 2 little b's. To work this out, we would draw a Punnett square. So the options for brown eyes, mum could have been big B, or she could have been big B, little b. Dad could have been big B, or it could have been big B, little b. So if we draw a Punnett square, and we have mum here, big B, little b, dad here, big B, little b, and we fill it in, and circle the one that would give a recessive condition, this one would be the baby with blue eyes. So we can summarise from this that mum's genotype was big B, little b, and so was dad's genotype. That isn't finished though, guys. You must give a percentage. So it's 25% chance of having blue eyes, and you can say 75% chance of having brown. Okay? Also represents a 3-to-1 ratio. Okay? So that's just the basics of inheritance. You also need to know a little bit about inherited disorders and sex determination. So we'll do sex determination first. So males should be XY, that'll give you a male. Females, their sex chromosomes are XX. This will always result in a 50% chance of having a male, 50% chance of having a female. Because if we split it, the gametes that could be produced by males are always X or Y. Females, however, can only ever make a gamete that carries an X. So if we were to fill this in, then 50% of the offspring would be male and 50% would be female. Okay? That's sex determination. That's more likely to come on a foundation paper rather than a higher paper. The other bit about inheritance before we do a couple of exam questions, you need to know about the inherited disorders. So there are two. There's cystic fibrosis and there's polydactyly. Polydactyly is the dominant condition. It's carried by or caused by a dominant allele, so you only ever need to have one version of that allele present. It leads to extra fingers or sometimes toes. So if a person, I'm not going to use P because P looks pretty much the same either case, I'm going to use a D. If a person was big D, they're going to have polydactyly. If a person was big D, little d, because they still carry that dominant allele, they're still going to have polydactyly. But if a person was two little d's, they would not have polydactyly. That's your dominant condition. Don't be thrown in an exam though. If it gives you another condition like Huntington's or something like that, that is also caused by a dominant allele, so the same rules apply. The other one, we've got cystic fibrosis. This is caused by a recessive allele. And you can identify an allele as recessive because if you look on a family tree diagram, you could have someone showing a cystic fibrosis even though neither of their parents had the actual cystic fibrosis condition. They could be carriers, okay? So if we look at this, cystic fibrosis is a disorder of the cell membrane. It has asked that before. and it leads to thick mucus, which can affect the lungs, it can affect the respiratory system, it can affect the pancreas. If it affects the pancreas, a person with cystic fibrosis would show poor growth. This is because the mucus blocks the duct, so enzymes can't get out to where they need to, so food cannot be digested. If we want to look at the alleles for this, I usually use an A because the C doesn't look any different in either case. Big A, big A, big A, little a, and two little a's. Because it is a recessive condition, it would be the one that has two little a's that has cystic fibrosis. Big A, little a, we will call a carrier. Two big A's will be unaffected. Okay, so that's a little bit about inheritance. I'm going to get the exam question in front of me. We'll save these sheets for you guys so that if you want to have a go at them, then you can. Or look at them when we stick them up on canteen window. So we've got an exam question. Polydactyly is an inherited condition caused by a dominant allele. The figure below shows a hand of a man with polydactyly. The man has an extra finger on each hand. The man's mother also has polydactyly, but his father does not. So if we have a look, it tells us it is caused by a dominant allele. It says the mother has polydactyly, but the father does not. If it is a dominant allele, then the father not having it must mean that the father is recessive, so that would be two small d's. Mother, we're not really sure. She could be big D, or she could be two little d's. No, she couldn't. She could be a big D and a little d. That's me going wrong there. The man is heterozygous for polydactyly. Heterozygous means that he has two different alleles, so he's a big D and a little d. Explain how the information given above shows that the man is heterozygous for polydactyly. Well, if we have a look, where has he got this little d from? It must be coming from his dad. Where's he got the polydactyly allele from? It must be from his mum. It doesn't matter whether she's that or that. So you get a mark for saying that the dominant allele came from mum. The recessive allele was from dad. And the reason we know this is because dad did not have any dominant alleles. And that would get you 3 marks. Then, if we move up a bit, the man marries a woman who does not have polydactyly. What is the probability that their first child will have polydactyly? So if we look up here, the dad is heterozygous, the mum doesn't have it, so the cross, if we needed to do it, would be big D, little d against two little d's. If you draw your Punnett square, you wouldn't need to label it up because it's only worth 1 mark. You should find that the ratio is 50% chance. So fill it in. Having polydactyly, it is 50% chance. And that's what you'd write there. 50% chance. Okay? That's how you'd work that one out. Then, very difficult question when it came up that threw quite a few people. It's about hair colour. So, the man has red hair, his sister has brown hair. Both of the parents have brown hair. So, man's got red, sister has brown, both parents have brown. Brown hair is caused by a dominant allele, red hair is caused by a recessive allele. Complete the genetic diagram below to show how the man's parents were able to have some children with red hair and some with brown. If two people have managed to have children with different hair colour to themselves, you would assume that they are heterozygous. So if you'll have a look here, if red hair is recessive, that must be the 2 lowercase alleles, so that must be BB. Brown hair, you could have brown hair if you were big B, or big B, little b. If we have a look at this quickly, because I know I'm going to be short on time, parental phenotype, that is a physical characteristic. Both of their parents have brown hair. So their phenotype, they have both got brown hair. Brown hair. Their genotype or their genetic makeup. If they have managed to have a child with red hair, which they have, there's absolutely no way that they could be that, because where would that recessive allele come from? So their genotypes must be big B, little b. This is a bit that's thrown quite a lot of you when we've done this in lessons. When it asks you for the gametes, all you're really doing is splitting apart the genotype. So they could make a gamete that carries a big B, or we could make a gamete that carries a little b, and it is the same for both of them. Then there's a space. If it ever gives you a space on a genetics question, it's probably wanting you to draw a Punnett square. So it's just like a multiplication grid. Again, it's very unlikely that you'll be asked to draw your own for foundation. This is more for higher. You'll be given a grid to fill in for foundation. So we circle the recessive one. We would say that that one has red hair, and we would say the chance, 25%. If it says what are the offspring genotypes, it means what options for the letters. Well, we've got big B, we've got big B, little b, we've got that twice. I'll just write it once. We've got two little b's. What would the phenotype or physical characteristic be? Well, those two, because they're both dominant, would give us brown hair. This one would give us red hair. And that will get you a total of 5 marks. They are big questions. Okay? Right. I'm going to move on. I'm going to skip out a few exam questions. So I really want to talk to you about, I've just thrown them all away, it would appear. Evolution and natural selection. So, please do not confuse these terms. So when we're talking about evolution, we mean that the theory, no, I've not had a capital letter, the theory that organisms have developed from simple life forms that lived billions of years ago. That is the theory of evolution. We think that this occurred, or we hypothesize this occurred through natural selection. So natural selection, the idea of it was mainly, for what you need to know, put forward by Charles Darwin. And he put together the idea that within any population there was variation. So if you get a question like this about natural selection, the first thing that you would need to mention would be that there was variation or differences in a population due to random mutations. It even rhymes. So there's variation in a population due to random mutations. In these populations, it could be of plants and animals, there are far more young that are made than can survive. So, unfortunately, there is competition, or fortunately for natural selection, there's competition for resources such as food, mates, territory or space. The ones that are best suited, or the best suited phenotypes, survive, they breed, and they pass on their genes. That is really important. It's also important that we say random mutations. It's not something that's thought about. You need to start trying to use the words best suited. You'll probably still get marks for best adapted, but the word the spec wants you to use is best suited. The only thing I want to stress here, guys, is that you must apply it to the scenario. So if it's talking to you about tigers and stripes, you need to say what the variation would be. So if we were looking at how the tiger developed stripes, there would have been a random mutation which made some tigers stripy and others weren't, would have been competition for food. The best suited ones would have been the tigers with stripes because they would have been better camouflaged in the forest to catch their food. These survivors would then breed and pass on their stripy gene. So you must use a term or what we're looking at. We're going to look at an example now, very, very quickly. Can I have 2 more minutes, Miss Froggatt? Thank you. So this is a hard one. It's one of the hardest ones I could find. I can't even say it. Soay sheep lived in the wild on an island off the north coast of Scotland. No people live on the island. It's a nice little sheep. It's got some horns. Pretty standard actually. Over the last 25 years, the average height and mass of the Soay sheep has decreased. The scientists think that climate change might have affected the size of a sheep. Well, what's happened to the size? It has got smaller. What has happened to the climate? It has got warmer. Suggest an explanation for the evolution of the wild Soay sheep over the last 25 years. Well, I dislike the term evolution in 25 years because it's a very short time span. But anyway, the first thing that we would need to say when we talk about this would be the variation. So there would be a mutation. Excuse me, guys, I'm sniffling. That leads to variation in size. Apologies about the focus, hopefully it's better now. So there's a mutation that leads to the variation in size of the sheep. I would link it to the context, so I would say that some are small and some are big. Then I would talk about it being warmer now, because it hasn't said it there, I'd probably get a mark for it. Climate change has made it warmer. This means that the smaller sheep are able to survive over winter because they wouldn't normally. Then the small sheep breed and pass on genes. Okay? So that is how you would apply that to the context that you have been given. I did have 2 more sections to go through. I'm a little bit conscious of time. I'm just going to sneak 1 more section in, if that's okay. Okay? I'm going to leave fossils and extinction, guys, because it says, all right, it's realistically nice and easy. You should be fine on how a fossil is formed and how things go extinct. It's just a process of genetic engineering that I want to talk to you about. So I put this blank piece of paper up, but realistically, all we're going to get a chance to do is the exam question. So if we have a look at it, insect pests can be controlled without using chemical insecticides. The issue with chemical insecticides is that they build up in food chains. And they can also be toxic to us. And it says, for example, and I love this because I can't ever say it, I don't know why I've picked this question. The bacterium Bacillus thuringiensis produces a toxin extremely poisonous to certain species of insects. The gene which produces this toxin has been introduced to tomato plants. It gives them built-in resistance to a range of insect pests, but is not poisonous to humans. Explain step by step how the tomato plant has been made resistant to some insect pests. Whenever you get a question about genetic engineering, you need to say where the gene is coming from and where the gene is going. It's easy to look at where it is going. It is going into the tomato plant. So this is where the gene goes in. Where is the gene being taken from? It's being taken from the bacterium. So it comes out of the bacterium. Okay? So with this question, the key points that we would need to make is where exactly in the bacteria does this gene come from? Because if you were to say the gene is taken out the nucleus of the bacterium, what you have said is absolutely scientifically incorrect and you'd be penalised for that. What you'd need to say is that the toxin gene is cut out from the bacterial DNA. Because we are scientists and we should be saying how, we use a restriction enzyme. Okay? This restriction enzyme will leave ends that are able to join to the tomato plant DNA. Once we've got the gene, this is transferred to the tomato DNA, which has been cut open with the same restriction enzyme. This needs to be done at an early stage of development, because if not, you would have to change every single gene in the tomato plant. So it's usually done when the tomato plant is still a seed. If you are genetically modifying an animal, it needs to be done when the animal is an embryo. If you are genetically modifying bacteria, it can be done at any point, and that's where you would use a plasmid. Okay? Your final mark is for saying, what is the point of this? Because it's an explain one. This means that the tomato plant can now make the toxin. So this tomato plant, if it is bitten by an insect or whatever, it will make this toxin, it'll kill the insect, but it's absolutely fine for us to use. I'm going to leave you with this little question, just whilst Miss prepares to take over from me. 2 arguments for and 2 arguments against controlling insect pests. These are the arguments for and against using genetic engineering. Okay, I'm not going to answer these. I'm going to leave and go over to Miss Froggett, who's going to take over and look at Spec 7. Thank you very much, Year 11. I'll be back at approximately 7:05 for separates only content. Thank you.
Right then, Year 11, we're going to have a look at Spec 7. And it's split. We're going to split it into 2 parts, interdependence and then environmental change. Now, interdependence comes in 4 different sections, so ecosystems, competition, adaptations and food chains. So we're going to start with ecosystems. Just switch pen. And this falls into community and environment. Now, a community is the population of different organisms in the environment and different species in their habitat. Now, their habitat is the area where they live. Now, this also links to environment, because where they live is their environment, but environment also includes, in terms of ecosystems, the conditions there. So what it's like, the weather, the climate, the levels of pollution, and these can be split into 2 different categories. So we have abiotic and biotic. So abiotic is non-living things that affect the environment. So these are things like light intensity, water, pH, minerals for plants, and oxygen levels for aquatic organisms. Biotic factors are living factors that affect a population of different species, and these link into both competition and food chains. So these are food and resources, predators, particularly new ones introduced, pathogens. So there we have our link to paper 1 in pathogens, and competition between different organisms. I too, miss, I'm having problems spelling competition. Now, that is a direct link to the next part in the spec. So animals and plants compete with each other, and within their species and with different species in their environment. Animals for food, mates, and territory. And plants for much more abiotic factors like light, minerals, and water. And again, we have another link back there to conditions needed for photosynthesis. Now, competition changes the number of organisms within a population, and that links to which organisms are best suited and which organisms are better competitors are the ones that are adapted. Now, the adaptations fall into two types, really. They fall into hot climate and cold climate. Now, cold climate, they're the ones at the Yorkshire Wildlife Park, the polar bears. I won't subject you to a drawing of a polar bear. They have thick fur. They have large feet. So thick fur to stop for insulation and to trap heat between the layers. Large feet, so them falling into the snow, spread the surface area. So we get within this another link to surface area for those people that hated the cubes in the first paper. They're camouflaged. They have small ears to reduce heat loss. You have to tell me if it's getting a little bit messy. To reduce heat loss. And they have, and this is the one linked to the cube, so there's no reason why tomorrow the cubes couldn't come back up where you're having to calculate surface area to volume ratio. Now, for the hot climate, we're looking at camels, and we give an example of a plant in cactuses as well. Now, camels have thin fur, because they don't need to trap as many layers of heat. They can tolerate high temperatures, and they have a hump that is a fat store. So that's their adaptations. They also sometimes link elephants' ears to hot climates as well. So that idea that they have large ears, they can lose a lot of heat through their good blood supply, all that kind of stuff. They also, like cacti, they have a large root system. They're often interconnected, the root system, so that they can pass water between them as well. They have a fleshy body, and that's the term they're tending to go for, is a fleshy body that stores water. They also could link back here to transpiration and reducing water loss in plants from paper 1. So they're still expecting you to remember everything that you'd already learnt, not forgetting it. And then we look at food chains. Now, food chains, sometimes they ask you to fill bits in on food chains. Sometimes they ask you to draw them. Sometimes they link it to energy. So that's what we're going to look at. Now, we're going to have a really simple food chain. We're going to have grass. We're going to have a rabbit. And we're going to have a fox. Now, in food chains, we're looking at the energy. The arrows represent the energy transferring from one organism to the next. It's not who eats who, it's where the energy goes. Now, we also have some key terms in this as well. So the grass is a producer. As it produces its own food, another link here to photosynthesis. Then we have consumer. This is a primary consumer. And then we have a secondary consumer. Within this, we also have the terminology of predator and prey as well. So we have a rabbit being prey and the fox being a predator. And as the food chain increases, the energy pass between each one does reduce, because not all of the rabbit is transferred energy into the fox. There will be some left behind in the bits the fox couldn't eat. Now, this also links into the carbon cycle as well, when we come to that in the next bit. Now, because this is all about interdependence, they occasionally show you a predator-prey cycle, which will be a graph, and it will be a graph of population against time usually, and it should show you the prey cycling, so the population of the prey going up and down. Not sure quite what's happened in that tiny bit of it. Then we get the predator's population. Now, I'll put prey on that bit so you know. And it shows you as the prey's population increases, that has an impact on the predator's population. As there's more food to eat, they're more likely to survive, so their population increases. That then affects, again, this idea of affecting within an ecosystem, the prey population starts to fall and it becomes a cycle, hence the predator-prey cycle. The other thing they like to look at in this is field work. So I'm going to show you an exam question. And it's a particularly unpleasant graph. I thought we'd pick this evening. Bit of an unpleasant graph, so can we see that? Now, what we've got is a coastal habitat. So they quite like the idea of the seaside and investigating organisms at the seaside. So we've got a coastal habitat. So the first thing is, that's where we are. We're at the seaside, and the picture shows the sea there and the shoreline. It also includes animals that, you might be out rock pooling, collecting animals like this. You might not be. Just be aware that any words that you don't understand in these questions, limpets, barnacles, periwinkles, they're the organisms. Okay, so they're what we're looking at. Now it says that the limpets, barnacles and periwinkles are animals that live on rocky shores. Students investigated the distribution of these animals. So how many there is and where they are. So that's what we're looking at. So the amount and where they are on the shoreline. Now, here we have on the graph the 3 different types of organisms, and at the bottom it shows us the distance from the shore. So they've gone out 6 metres. And what we have here is a kite graph. Now, if you look at the bottom, shove it up a little bit, it gives you a key. If the exam questions give you a key, you must look at the key and work out what is the graph actually showing you. Now, it tells you quite clearly there that for every 5 squares, there are 10 individuals. So that means for every 1 square on this graph paper, that is 2 different individuals. So that means 2 different either limpets, barnacles, or periwinkles. And the first question is to calculate. Now it says, the edge of the sea was 6 metres from the top of the shore, and it says calculate the total number of animals found 1 metre. So we need to look at the graph and find 1 metre, which is here. And just like you would do with any other type of graph, you go backwards up the graph to here and here. So we've got no periwinkles, but we've got some barnacles and limpets. Now this bit, if we go across, is 1 square in width. So we have 1 barnacle. Now we know from before, from our key, that we need to double the number because 1 square is 2 individuals. So we have 1 square, which is 2 barnacles. Then the number of limpets, we have 2 squares worth of limpets. So that is 4, because we know 2 squares is, 1 square is 2 individuals, so we have 4 limpets. We literally add them together and we get 6. That question caught a lot of people out. They were put off by the graph, but actually, not only the science, but the maths is quite easy in that. And if we have a little look, we get our link. It asks, we're going to concentrate on this top part of the question, which is describe the methods the students could have used to collect data from this figure. So we'll put our figure back on a little bit. Now, in this question, they're asking you to determine how to collect the data. So the first thing we need to do anywhere where we're looking at living organisms in their environment is select a location. Now, the other thing that keeps coming up in questions is select a random location. You don't have to be specific, you don't have to say a number grid from Google Maps or anything like that. It just has to be a random, even if it's just you pointing at the seaside and picking somewhere. Yeah, so select a random location. Then you're going to use a transect. Now, a transect or a tape measure, it says, to measure the distance from the shoreline. Each one of these bullet points in the mark scheme is a mark, and it's for 4 marks, so it's worth having a good go at it. So it says select a tape measure to measure the shoreline. And you can show from here, just like they did, every metre or so down the shoreline. And they don't really ask you to be, that's not a very good thing, you to be specific. Yeah, and it would say, every metre, can we still see? I'll shove it up a little bit. Every metre through, the other thing they're always looking for in these questions, the dreaded quadrat. So you will have had these out at some point, and they're squares like that. They've got squares in between. You throw it, you select a square and count the organisms within that square. Now, the important thing about throwing the quadrat, in order to count the species, you have to do this randomly as well. There's no point putting it down on the same bit of transect every time. You have to stand on it and throw it randomly. Then, just like with any good experiment, you need to repeat it. Now, they would accept, at this one, repeat it at different locations on the same beach, or they would also have accepted different times of year or different seasons, because that would obviously affect the number of organisms on their temperature. Now, we're going to look at the last part in interdependence now, which is environmental change. Now, this is the one that links into the chemistry as well, because it has global warming on and humans' impact on the environment. So, the first thing we're going to look at is natural cycling of materials. So, ones where naturally materials cycle, and we're going to look at the water cycle, and we're going to look at the carbon cycle. So, those are the two that you need for this one. Now, we'll start with the water cycle. Switch that out. Now, the water cycle you will have learnt at some point in primary school, and we keep coming back to it as it's really important, particularly for if we're linking into global warming, we're looking at maintaining diversity in the environment. Now, if we start with the oceans, the water's evaporated from the oceans, it goes up as water vapour into the clouds. Then it begins to cool, we get precipitation. Some of this precipitation falls on land, some of it trickles through, not sure how well you can see that, into the soil. Then we get a link here in this part back to paper 1, because you have studied in paper 1 transpiration and transporting plants, so transporting water from the roots, and you've looked at respiration as a way of releasing water vapour into the environment. And you will notice in this diagram it says from plants and animals. Okay, so that loss of water through transpiration from plants, but also through respiration. Now, the other way of naturally cycling in the environment is carbon. Now, the carbon cycle is, if we look, this interesting link between, here we have fossil fuels, we have animals in the middle, and we have plants on the edge there. Now, we also have, I don't know how clear that is, so I'll write it above, decomposers. Now, what we have to look at is where the carbon goes. So we have carbon in the air, in the atmosphere, Now, all the links to carbon in the atmosphere, we'll look where they come from. So we have a link from animals, we have a link from plants, and we have a link from decomposers. Oops. That link there is respiration. So I'll put a little up there. That is respiration. So not only are we respiring, plants are respiring, decomposers are releasing carbon dioxide as well. Now, if we look at the transfers within the cycle, we'll have a little look at decomposers. So plants decomposing, animals decomposing, the carbon from them going into decomposers, those respiring, releasing it into the air. We also have a link between plants and animals, because we eat plants, animals eat plants, so the carbon from those goes into the animals, and they are the cycles within there. Now, this one links into global warming and into the chemistry, because this one is where we get carbon trapped in the environment, for it all the way around, for millions of years. Now, plants, they can become fossil fuels, like coal. Animals, they can become oil and gas. Now, this one traps carbon for millions of years. And it's one of the main ways that we can reduce carbon in the environment. But obviously, we are releasing carbon from fossil fuels into the environment through combustion. So that's what you can see there. Now, that carbon has been trapped there for millions of years. We're combusting fossil fuels and releasing it back in. Now, one thing you will notice from this is every arrow so far has been ultimately leading to carbon dioxide in the air, increasing the levels of CO2 in our atmosphere. There is only one arrow that shows carbon coming out of the atmosphere and being trapped. And that is from photosynthesis. So if I highlight that bit, from photosynthesis. Now, this bit links into deforestation and global warming that we're going to go on to now, and humans impacting the environment. So this is a natural cycle. It's been happening for millions of years, but now we need to look at applying that to how humans kind of mess this up now. Now, let's have a little look. So, we go back to our natural cycles. On the right sheet. Now, what we've got is we're going to look at humans' impact on the environment. So, really, the Spec breaks this down into pollution and deforestation. Now, pollution is usually quite well answered, but they're starting to be specific about land, air, and water pollution. Now, land pollution, they will accept landfills or dumping rubbish. They'll also accept chemicals. For air pollution, they're looking at smoke and particulates in the air, or the quality of air, and they're also looking at sulfur dioxide. And that links into our greenhouse gases, it links into global warming. For water, they're looking at sewage, they're looking at fertilisers, so runoff from farming, and chemicals, again. That could be from industry, could be from farms, it could be from households. So humans impacting the environment by adding more of these things into the environment than would have naturally been there in the first place. Now, deforestation, we have to look at the reasons human beings need to cut down trees, and then we have to look at the problems that this causes. Now, this does link straight back into that carbon cycle again, because we need to cut down trees to grow crops. Now, these could be for food, but they could also be for biofuels. We also need it for grassland, for cattle, and for housing, roads, infrastructure. But that does cause us problems because with that carbon cycle again, it increases the CO2 levels because there are less plants to trap that carbon. And it also means that we get floods and landslides like have been happening in America recently because they're cutting down their forests. There's no roots to hold the soil or water, and it's flooding into these areas that we have housing, cattle and crops. It also links to peat bog destruction, which is causing the fires on the moorlands that you might have seen on the news because they are a natural carbon deposit and they hold a lot of water. So again, it links to floods and landslides. Now, global warming, if you do geography as well, you've probably done this over and over again, but we are still looking at it in both the chemistry and the biology. But we've got it's caused by increased CO2 levels. So they could well ask you a carbon cycle question and then ask you to apply it to global warming and human impact. So we're also looking at the greenhouse effect. Now this is usually confused. So global warming is the temperature increase due to the CO2 levels. The greenhouse effect is the heating of the earth through methane gases and trapping heat. Now it's the impacts that come up in this spec. And that is the destruction of habitats, of animals. We've got rising sea levels, from melting ice caps. We have climate change. Again, not to be confused with global warming. Climate change is long-term changes in the climate and weather systems. It can also lead to floods and droughts. And that combined with deforestation is obviously why some areas are facing problems like that. It can also link into maintaining biodiversity, which is our last one, because changes in the environment and changes in climate and rising sea levels means that the animal population is at risk, particularly migration patterns and things from birds. Now, what we can do, now we've done all these things to the environment, what we can do to try and maintain biodiversity in areas is conservation and breeding programs. Some people tend to put stop hunting when they're faced with a question like this. They're not really interested in that. They're interested in conservation and breeding. Okay, so improving the population of animals or regenerating their environment and their habitats. And that links all the way back to ecosystems and looking at the conditions in their environment and what they are competing for. Now, the three environments that they look at are wetlands, hedgerows, and forests. Now, they can ask you any animals in there that live in environments, hedgerows, things like hedgehogs, wetlands, birds, marshlands. You don't have to have studied the exact animal, but you have to be able to apply your knowledge to that. So hopefully that's helped out a bit, Miss.
Thank you very much, Mrs. Foggett. Right guys, that's the trilogy content over with. We're going to take a very short break and then afterwards we'll start a new live stream for the separates only. We are running behind, apologies, but we wanted to get as much done as we could. So if you are a trilogy science student, you do not need to watch the next video. If you are a separate science student, please stay tuned or make sure you come back online, whatever, I don't know what I mean. We'll be coming back with another live stream. Thank you.